to the weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 81st episode of the weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years now. My name is Dominic Guarino, and I've been in the bioanalytical ligand binding assay and biomarker space for over 31 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LC-MS space for around 30 years.
John and I are members of our growing scientific advisory team here at KCAS Bio. Either or both of us are available to answer any of your questions you may have regarding this podcast or any of KCAS services. We are delighted to have you here in the early days of fall, listening to the 81st episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're delighted to have you here, and we are looking forward to a fun episode today. To kick us off for podcast number 81, John is going to go over today's podcast topics a bit before we jump into the news and resources. John?
Sure. Well, as usual, we'll touch on some news items before we move into our main topic, which is revisiting GLP-1 Analogue development and bioanalysis, the value of complementary technologies. GLP-1s are everywhere in the news at the moment, and obviously there's a lot going on in that space within the pharmaceutical industry, and that has a knock-on effect to us. So it's just talking about what's out there, how we have worked with GLP-1s in the past, and some of the issues we're facing as we continue to work with GLP-1s.
All right, John, let's just hop right in. What's the first news article?
So first one is once promising cancer drugs appear to protect mice from Alzheimer's. And I mean, Alzheimer's, I think, will increasingly come up in this podcast simply because it's one of those high-profile diseases that we're still trying to figure out the mechanisms of it, etc. And this might be a step towards understanding what's going on. Researchers at Stanford University discovered a new role for metabolic enzyme IDO1 in Alzheimer's disease and found that blocking the enzyme with a cancer drug improved brain metabolism and memory in mice. This was published in Science, and it's again, we're just like I say, we're trying to get an understanding of Alzheimer's, and there's a lot of focus on amyloid and tau, but there's also people are thinking it because the drugs that have shown promise against amyloid and tau, their benefits are still sort of not really sure if there's clinical benefit from the drugs, even though they've shown reduction in amyloid and tau. So that's what still says, is there a question over what's actually going on? And the Stanford group were looking at IDO1 inhibitors that were initially thought that they could be useful in cancer, because IDO1 makes kynurenine, which helps shut down immune responses. However, tumor cells can hijack the process and produce more kynurenine to stop immune cells from attacking. And so the thought was that if you blocked IDO1, then you could help immunotherapies function better. However, when it came to cancer, that wasn't actually successful. This group at Stanford have actually shown that the enzyme, they got some additional insight into the enzyme and kynurenine in the brain, and so it's like kynurenine is actually destructive in the brain, and it can shut down glucose metabolism in astrocytes, so it then prevents them feeding neurons downstream lactate that neurons need. So it's the theory is that actually IDO1 is much more involved in this, and as they talk about it, they did some work in both mouse and monkeys, showed that they could actually help the mouse and monkeys, and it did have effect on both tau and amyloid, and so it seems that all are interlinked. So it seems to be part, rather than being just like a observational thing. It's actually intimately involved in the whole Alzheimer's process.
Yeah, it's really starts with it's a small molecule inhibitor, right, John?
Mm-hmm.
We kind of dust it off the shelf here, and it just happens to pass the blood-brain barrier. That's the first thing that
Yes.
made this thing attractive, right? And then you mentioned astrocytes. That's the cell type, which they're a little understudied, John. We do have, I think we've got us. I feel like we've done some flow work on astrocytes, but they're hard to study, right? Especially in a human setting. I mean, I think we've done some preclinical models, but it's not really something that you're going to go get a lobotomy, right? That's what that would be in order to do that. So, but it's one of those things where mechanistically it makes sense, right? This kind of chokes off the tau is kind of how they describe it. So you can think about reducing that, but is tau's healthy too? So it's a fine line. And man, all this, it's unfortunate that over the last, certainly the 4 years now we've been doing this podcast, a lot of Alzheimer's promising drugs have just kind of poo-pooed out, right? That's a technical term I learned in grad school, John. And so, even the last bit of this, it's like, yeah, we'd like to see it in the clinic. So I don't know if I'm just maybe in a little bit of a foul mood here early in fall, but unfortunately, I know this looks promising, and certainly, measuring these biomarkers, certainly it's just hard to think that we're going to unlock some of this. And I was even saying to somebody, the best way to present for me personally, I do more puzzles, crosswords, just keep exercising your brain, John. I guess that's still maybe the best thing for Alzheimer's, but
I think you got to realize though, mice aren't very good with doing puzzles.
Well, that's true. But they still maybe yeah. But no, there's a thing. It's a. I don't want to downplay this. It's a science publication. Fact.
Sure, absolutely, yeah.
And the way they did it is very eloquent. This is like cutting edge stuff. Mm-hmm. It's just Alzheimer's is just tough.
That's the problem with why we keep talking about Alzheimer's, is that huge question mark over it. It's still trying to figure out the mechanisms, how to address it. We're still looking at potential other pathways to attack it. This is one of them, and it's like, so the group at Stanford, it was actually an old Pfizer drug is where they actually saw initial success, but are looking at other small molecules to see what they can do. And they say this drug restored metabolism and memory impairments in mice. So it's a way forward. I mean, I like, with this, I'm constantly looking for articles, and we're getting prepared for podcast number 82, and I've already got one that I will probably end up talking about.
Yeah.
And it's another mechanism for possibly going at these protein accumulations in Alzheimer's, and that's obviously next time. We're not touching on it this time.
Yeah, no, I think you bring up a good point, John. It is such a thing that we're all wanting to see.
Yeah. I mean,
I love the story too. Repurposing a drug is such a powerful thing.
Yeah, as we get older and we know more and more people affected by Alzheimer's, I doubt there's any of us don't know anyone either in family or friends who haven't been affected by this. That you want to see some progression, and it's a slow process at the moment. We want to. We'll keep coming back to it because you want to have a message of hope on this stuff. But to me, some of these like ALS and Parkinson's and Alzheimer's, they're all the ones where you see companies involved in this. You think that the reward could be huge, but the risk is huge as well, as we try and figure out.
And it's even the last piece of the parts of the article talk about how they want to enhance this. It's cool because the blood-brain barrier is still a little bit. There's not controversy, but they thought it was just like kind of like really a rigid thing when it's not. There's probably more active versus passive things going through the brain, John. But it seems like this is pretty cool. I guess as I kind of dig in, and maybe you're just cheering me up, John. But let's move on to the next article.
Well, the next thing is actually it also sort of plays into this because this I tend not to focus on commercial companies when we're doing this, but this is Denali, who we know have been involved in the central nervous system type of diseases, and they report a new method to get antisense oligonucleotides which across the blood-brain barrier. So basically, these ASOs bind to RNA, prevent it from being translated into proteins. There's a lot of interest in this area, particularly for treating neurodegenerative diseases. However, the one of the barriers has been, or one of the hindrances has been getting these drugs into the brain. So, you don't have that so when it comes to administration, you might be doing it intrathecally, and that comes with its own complications. So if you've got a mechanism whereby you can get these larger molecules that could be effective in the brain across the barrier, then you've got another way you can actually dose intravenously, which is the point that's been made in this article. And they actually, Denali did this in studies with mice and macaques, and this is where I misspoke on that previous article. It was only in mice. I was conflating the two. This is in mice and monkeys, and the actual means of transporting the ASOs is a transferrin targeting transport vehicle platform. And that so basically, they were able to get brain uptake of the ASOs by using this delivery system.
Yeah, it's a big advancement, right? So
Yeah.
they even talk about how intrathecal administrations require a large bolus of the drug, right? And this
Yep.
and this can avoid all that. And they talk about biodistribution studies, near and dear to our heart, John, right? Really cool stuff to be able to take advantage of what's known, as I was kind of stumbled across, right? There is passive and active ways to get across the blood-brain barrier, and certainly the transferrin's one. We know that the brain needs that. So taking advantage of biology there and nature and getting their ASO across. These ASOs are wonderful drugs, in my opinion.
So yeah,
they're solving a lot of problems, and to be able to enhance them is awesome. Sorry, go ahead.
So just a little bit of background. It says Denali's transport vehicle platform is an engineered antibody that targets transferrin receptor 1 across the barrier.
There you go.
So that's that. So basically, that's the technology, how they're driving this, and they did like a proof of concept, focusing on a gene called MALAT1, but like say.
So they're getting a biologic across the blood-brain barrier, right, John? Yeah, that is exactly right. That's what they're doing. They're binding.
Yeah.
It's basically it sounds like it's almost like an arc, right? Like it's an antibody. Or am I mistaken here?
Well, I don't know. It's
not an arc necessarily, but they're using. Go ahead. Sorry. Go ahead.
Yeah, I don't know what the form of binding is. I don't know if is it a true chemical binding that then releases it, or I don't know what it is. Is like is it anomalous to like a LNP type?
Yeah, I don't think we're going to solve it. That's probably their IP. So I don't feel bad. This is
they do say that binding an antisense oligonucleotide targeting MALAT1 to Denali's transport vehicle forms an oligonucleotide transport vehicle (OTV), which allows the ASO to be shuttled into diverse cells and can successfully reduce expression of the gene in both mice and cynomolgus macaques. So it does sound like it's a chemical thing that then it breaks once it's in the brain. But yeah, like I say, the advantage is it's getting these large molecules across the barrier that is normally resistant, and then, like I say, downstream makes possible treatment for patients much simpler and more user friendly, because it's just an intravenous drip, and like I say, avoids that bolus of dose and things like that. So it could be a safer approach and everything else. So it's a really, like I say, I just thought nice, elegant approach, and could have a lot of benefit downstream.
Yeah, and they target all the, it's interesting. It does parlay nicely in with the IDO1 article previously, because they look at alpha synucleins and tau and all that. I'm sure they got their eye on this IDO1 now, right, John? Like it would make sense to be therapeutically treating that. But it's really cool stuff to be able to take a biologic, target the surface, and deliver a drug via a biologic to the brain. That's wild to me. I guess I think that's pretty cool science, and hats off to the Denali team for that.
Yeah, so basically, Denali intends to seek accelerated approval for DNL 310, which is their ASO in development right now, and it's in phase 3. It's an enzyme-based lead candidate for Hunter syndrome, which I assume is probably like a rare disease. But
Yeah, really
yes,
impressive.
let's move on to
Yeah.
our next one. This is one that I have read multiple times, and I'll be absolutely honest, I'm still struggling to quite get my head around what's going on. But it's fascinating, and this is actually trying to dig into the mechanisms of what spurs cancer or helps the propagation of cancer. And the headline is Mitochondrial Free Radicals Spur Micronuclear Collapse in Cancer. This is actually it's interesting. It's two papers talking about the similar stuff. One is from the European Institute of Oncology and the University of Milan, and I'm not sure where the other one's from. We'll no doubt touch on that. But basically, it's talking about the rupturing of micronuclei and how they collapse in cancer cells, which is an important part of cancer progression. The micronuclei get tangled with mitochondria. They're then exposed to reactive oxygen species that alter the behavior of a key repair protein and overactivate a protein destroying pathway. Now, I read this as like I said, the mechanism of all I'm still trying to figure out. But I do, once you start seeing about the protein that they talk is implicated on this called P62, and it gives an indication of a potentially future biomarker for looking at cancer patients, because they basically said that they looked at 875 gastric cancer patients and found that higher levels of P62 were correlated with shortened survival. So there's a biomarker in terms of to monitor in terms of obviously like pathway for the disease, etc. I'll pass this to you, and then we'll dig into it a bit more as I get my head around some of these pieces.
I love a good mitochondrial. If anybody's listening to the podcast,
like I say, it's fascinating, but it's because it's also dense in terms of what
it is. John, you're right. It is a molecular biologist dream here. Well, actually, I think it's a cell biology thing. But mitochondria have, to me, always been a way to prevent aging or help you stay young and maybe even fight cancer. And there's a lot of studies on this. But it seems like when your mitochondria go bad, it actually can cause a cancer, right? That's what this is saying. And it talks about a couple different proteins that literally tug on the membrane of the nucleus and cause it to cause the micronucleus membrane to collapse. And therefore, tumor cells become aneuploidy, right? They either have too much or have too many, and that might be the mechanism behind it. This could be revolutionary in terms of how to attack. It's at the most. Think about it. It's at one of the earliest stages when a cell could become tumorous, right? And also, it's one of those things that might be repairable. But again, I don't know how you can. It's not like a collapse. Well, I mean, they can get a collapsed lung to go up, right? But it's almost like this is collapsing the mitochondria, which is again just causes the whole thing to go haywire. So, John, P62 is a protein that I haven't studied a tremendous amount about, but that's the kind of the mechanism behind some of this. But these other proteins, certainly LEMD2, right, and the CHMP7, but the way they describe it is really fascinating, and it reminds me of some of the cell biology I studied where, I don't know how much you know about neuronal receptor channels, but they have like a ball and chain, and this chain will sit there and prevent, it's actually along the blood-brain barrier, it'll prevent things from falling in, and when people get a mutation, that ball doesn't place, and that can cause a tumor. How they even discovered this is fascinating, but my goodness, John, this could be revolutionary as far as I can.
Sure. Yeah. I read this article and thought, okay, this is absolutely, we have to talk about this because we can. Yeah, I don't even know quite where to go. It's basically. Yeah, because Melody DeBona was a cancer biologist working in Samuel Backham's group, and I assume this is at Milan. And she noticed that micronuclei tend to get tangled up with mitochondria. My hypothesis when I started to look at cells was mitochondria were actually entering the micronucleus. She was looking further into this, but she actually found that the chance of rupture greatly increased as the micronucleus was exposed to more and more toxic forms of oxygen-containing molecules leaking from the mitochondria, and so she sort of then figured out how this happens. So tumors tend to be low in oxygen, which the mitochondria needs to function. When oxygen is low, nitric oxide binds in its place instead, which produces these reactive oxygen species as a byproduct. These are unstable, can steal electrons from other molecules, messing with their form and function. She found that these reactive oxygen species took electrons away from cysteines in the protein CHMP7, changing its form so it abnormally binds to another protein called LEMD2 that sits inside the micronuclear membrane. What's more, these reactive oxygen species damage also prevents CHMP7 from being kicked out of the cell. And this is where you talked about the CHMP tugging at the LEMD2, which is there's a whole interaction.
There is, John. Then they talk about NF-kappa B, which is well studied, and that gets into a whole nother, kind of territory of just cell mechanisms. But
Sure.
It's HPV-7, which is, was it HPV cervical cancer that she studied? That was interesting. I don't know if you mentioned that. I found
no, I didn't. So
anyway,
this is one of those things you look at it and there's a lot being talked about here, and it's with this identifying this mechanism is another way that we can obviously look at some of these cancers and address them.
Yeah, maybe the next time someone asks me if there's a cure for cancer, I'm going to say there could be, because this and actually they talk about it being we couldn't suppress the instability. They want to like the cat's out of the bag, right? We can't undo it, but they could potentially like kneecap it. That's the word they use.
Yeah.
It's got kind of hard to envision how you treat systemically for all this, but you know. CRISPRs and mRNA vaccines are doing unbelievable things these days. So who knows what you can do to kind of gene edit, but that would require some interesting gene editing to kind of do this. But John, I think these are the types of things that lead to our grandchildren getting therapeutics, right? This is really cool.
I mean, I when you say can cancer be cured, I mean, I say not now, but the way medicine's progressing, at some point, yes, it will be.
Well, I mean, every day we're a step closer, right? There are a lot of cancers that are being cured, or at least, you're living a healthier, longer, normal life. That's the key piece.
Sure. Yeah.
But like leukemia, lymphomas, and lots of lung cancers and head and necks, all of that.
Yeah,
not a death sentence like it once was back in the 50s and 60s. That's
a lot of these things that were death sentences, there's a lot more hope to them.
There's absolutely. Still a couple tumor types that are, anything in the stomach or more systemic gets tougher to treat. But, ADCs, everything's working. All right, that was a little side bob there, John. I think
that's the news.
And John, we're going to move into our main topic, right? Like, it is such a great topic. I'm John, we're gonna revisit the GLP-1 analog development and bioanalysis. Right?
Yeah, it's like everyone else is talking about them. Why shouldn't we?
Yeah, the value of complementary technologies. And John, it's so. It's okay, John. I got pretty excited about this topic just from like the field is moving. It's moving so fast, John. Right? I feel like the GLP space. It's just because of its headlines, right, as well as even. This is not a new thing, right? This has been all in the, let's call it, diabetic space for a couple decades now. But now, John, talk to us about some of the latest developments, because these things are it's hard to keep track of there's so many people in the space. There's so many different types of it. There's so many little nuances. And now, John, there's the best part is gene therapy is arriving, right? But tell us a little bit about some of the latest developments in GLP.
Sure. Well, where are we going to start? That's an interesting one. So, I mean, I think first of all, we have to acknowledge the heavyweights in GLP-1, which are semaglutide in the Novo Nordisk camp, or Wegovy, or I can't remember the brand names. And the other corner, you've got Lilly with tirzepatide, which is Mounjaro, and it also has other brand names. But both those are diabetic medicines, both show benefits in weight loss, and they're both approved for both indications. But it's the interesting thing that the more they dig into these compounds, the more they're seeing other potential medical applications. And I mean, we talked about these in the past, but obviously there was like tirzepatide, the Lilly drug, there was talk about its potential benefit for sleep apnea. There's the cardiovascular studies that have been done with the Novo Nordisk drug. So there's a lot of focus on these ones. We've also, from the point of view, these are really interesting molecules, regardless of their medical application. Obviously, they've had that progression that I like, where there's a root of these in nature, where GLP-1, obviously, I can't remember the mechanism now, but it helps the GLP-1 helps with insulin management.
Yeah.
The issue with GLP-1 is a short half-life. So the first drug that was developed was actually similar to a compound in the venom of the Gila monster. No, the saliva of the Gila monster, sorry. Not, I'm obsessed with venoms because a lot of work I did in the past. Anyway, so in the saliva of the Gila monster. And so that was the first one to imitate GLP-1, and then it was timely, this, because actually there was one of the things that we should touch on is that the understanding of all the mechanism here, and just the drug design resulting from as a way to address the GLP-1s has actually led to a science prize being awarded to the principal scientist at Novo Nordisk. So anyway, structurally, like I say, with GLP-1 it's effective but short half-life. So for us, for the drug companies, the big deal is, how did you address the half-life in a biological system?
Yeah,
And the approach that was taken by Novo Nordisk, and I should try and pick up her name. I can't find where I don't seem to have it. Sorry. Anyway, the least at Novo Nordisk, they took the peptide and attached a fatty acid chain to it, and that fatty acid chain drastically altered the half-life of the drug, so it became much more applicable for addressing the diabetes, etc. That was a huge step forward, really, and that's where a lot of the drugs that have followed, and semaglutide wasn't the first one. It was like a second or third generation in line at Novo Nordisk, but it did address how to make these peptide drugs last longer in the body, and therefore have a therapeutic effect. So this first generation are really variations on a theme, where it's that peptide backbone with a lipid chain added to it to help the half-life, and then that's your basic drug design, which I'm sounding really simplistic. It's much more than that, but that's really the mechanism of these first generation of GLP-1s. But the drawback with these, where we talked earlier about intravenous and versus thecal, intravenous isn't necessarily convenient. So a lot of focus since then has now been on oral versions of these drugs, or oral drugs that will do the same job as semaglutide and tirzepatide. I'm going to pass it back to you because I've done a lot of talking. I need to get my brain unscrambled.
No, you actually. I'm just going to recap a little bit of that. Just if people aren't that familiar with this, that might be a little bit easier to just recap. There are two major players, Eli Lilly and Novo Nordisk. They each have their own type. They're given injectables. And when I say type, GLP-1's a hormone, right? It's like a peptide, right?
Yep.
And so
Absolutely.
each of them had these, and they were treating diabetic. And then they suddenly realized, oh my God, you can lose almost 20% of someone's weight with this. And it has mechanisms of actions through insulin that help. It actually really suppresses the desire to eat. And that's that. They're not 100% sure how that all works, but it is driven through like your insulin feedback loop, and it's all part of the axis loop in the brain. But again, I don't know how well that's studied, and I haven't studied it.
There's a couple of
But
there is some publications out there. There was one in Nature recently, and so that people are looking into this to figure out which part of the brain is being affected by it.
But John, these drugs they're like years old. They've been on the market for a while, a lot of them, right? And so they took them, and then what did they, just like you said, they added a fatty acid to make it more stable, right? So that went from, you might need an injection daily to weekly or something like that, right? And that's the evolution, and they got more specific, and there's a market in there, right? All how potent they are, and how there's all sorts of, even Lilly and Novo Nordisk are creating different iterations of them. And then now the field has moved into what? An oral form. So we just went over like 10 years of like GLP-1 pretty quickly. But and then John, I just want to cap it off because I think you're going to talk a little bit about the small molecule piece. Why don't you, I don't know if you want to move on to talking about the oral version, but I want the audience to know, look, the field has gone from like daily injectables, maybe a couple times a day, to weekly, even monthly, right? And now we're talking about a pill to eliminate that altogether. And then what's going to happen? There's all these gene therapies. I'll talk about those, John, but tell us, like, how's the small molecule? Because it's not oral format, isn't quite there yet, but I can't imagine it won't be, right?
So yeah, I mean, I think it's a case of right now, it's obviously diabetes is a chronic
40%,
condition that we're looking.
40% of the population is overweight. But that's a staggering number. That number is getting higher and higher. I just heard that this morning, about 40%.
So, but the big money pot in all this is the weight loss application, and so a lot of the focus, particularly for these like the oral drugs, as you're looking at these studies come through, a lot of the emphasis is on, even though they're in early development, how much weight loss they're seeing from subjects and how quickly in the process they're seeing a percentage of weight loss. So then it's like it's extrapolating how much they're going to see beyond that. So a lot of the emphasis right now is really saying, Wegovy, Manjaro, those are your two gold standards. It's how do we beat those? And if it's a weight loss, what do we have to show up front to show that we're equivalent? Obviously, if you've got an oral that's easier administration and it's got equivalent weight loss to the two heavyweights, then you're in really good shape for going forward. There are a lot of companies working on this, and a lot of companies publishing data quite on a routine basis. So, for example, Bioanalysis is a company who published something recently. Their small molecule reduced the weight of obese mice by 15% over a 28-day span. So they're still in preclinical, but it's obviously showing that their compound has legs. So they're moving into clinical trials next year. Let's see what else. I think Novo Nordisk have got oral form.
And Lilly, I think both of them are going to try, and then every major pharma has a version of oral. Pfizer, Roche, they're all trying to throw their hat into this.
Exactly. So it's where we thought that where there was the two heavyweights, there's a lot of other people in this field moving into it. So in terms of bioanalytical, and that's partly, I know we've touched on this.
Hang on, before we move there, there are the latest. Now there are mRNA versions of this, John, and that's targeting specific tissues to produce it.
Oh, really?
That's hot off the presses. That's why it's actually somebody we're working with, John. And so they're using an mRNA sort of gene therapeutic, and they're knocking it into a certain organ.
Oh, yeah, and I know there's the gene therapy out there.
Yeah, so
Been
that's
published
that's
out there.
a yeah. So
Yeah.
that just as a that's the next wave, I think. You got injectable to oral to like one shot in a gene
Sure.
therapy type thing, and we can move on. We can move on to the topic because I want to cover everything. But I think it's important to think about that. Just when we like, what's the latest developments in like this GLP-1 space? It's like, hey, we're getting injectables are going to be not a thing of the past, but maybe once a month or even maybe in every couple months or something. And then an oral version is going to be hot off the presses, which again opens things up. And then think about like maybe a one-time vaccination and you're good to go because you've knocked it into an organ so that it's producing GLP-1 at like a healthy level. Great
So,
stuff, right?
so on the oral front, I mentioned Bioanalysis, but there are others ahead of that. So Novo Nordisk have a oral amycretin, which they reported weight loss at 12 weeks at around 13.1%, but they're saying it hadn't plateaued at that point, so it's a case of watch this space as the study progresses. So yeah, like they say, there's an awful lot of focus in this area.
Yeah.
But ultimately, it comes back to while there's the drugs are coming through, there's also a body of or there's groups are looking at how do these things work. What's the? I mean, you mentioned that they one of the side effects is like these anti-obesity drugs. Yes, they help you lose weight, but they also cause intolerable nausea.
Yeah.
So there's one group at the Monell Chemical Senses Center in Philadelphia. They've pinpointed a brain pathway that is involved in this side effect, so that might change how they design weight loss drugs in the future to get away from that mechanism.
Change an amino acid, right, John? Is that what they're talking about? Mutating it a little bit?
They haven't said how they're going to do it. It's just that it's looking. It's very early, to be honest.
Well, that's some of the gene therapy people are looking at certain mutations to help with half-lives and things like that. So it's really, I wonder if that would be the way to, if they can determine what is causing it, then you can make a GLP-1 that doesn't, but still be active.
Exactly.
It's great science. Sorry, it is.
And that's
it's really cool.
So it's like they looked at, so just very quickly, they looked at two subregions of the hindbrain in mice called the area postrema and the nucleus of the solitary tract. When the scientists turned on the animal's AP neurons, the mice exhibited nausea and food aversion, reduced their food intake. By contrast, when they turned on the NTS neurons in mice, the animals cut back on their food but showed no signs of nausea. And that's because there's GLP-1 receptors in both those regions of the brain. And so it's trying to figure it out so you get to reach one set of GLP-1 receptors versus another, and that's how you hopefully, if you design the drug right, how you hopefully then control the nausea piece.
That's got to be a cool experiment to set up, John.
Oh yeah, absolutely, yes.
So like I think we've done a nice job of just going over the latest and greatest. Maybe we have more. Go ahead.
Just very quickly, actually, yeah. Before we can go into the bioanalytical and all that kind of stuff, because obviously that's how it impacts us, and we love this stuff. But actually, there's a group in South Korea, Seoul National University, who they identified brain area harboring two groups of neurons, which because they were trying to see how do these obesity drugs create a full feeling even before one bite of food. And so again, it was looking to two groups of neurons, and they found that one was responsible for pre-meal sensations of fullness and one for post-meal satiety. And let me see if I can. So they're still digging into the mechanism, but I love the fact that he and his colleagues recruited people with obesity and asked them to report their level of satiation at 3 stages: before exposure to food while looking at a delicious plate of Korean fried chicken, before eating it and after eating it. People taking liraglutide felt full even before exposure to food, but this feeling grew when they were shown food and again after they'd eaten. The findings demonstrate that this feeling full isn't just it's a widespread indication for people, but the people who hadn't taken liraglutide, they weren't the sight of the fried chicken didn't make them hungry, but they felt full after they'd eaten. So and they actually, for them, they honed in on the dorsomedial hypothalamus, which is GLP-1 receptors, and that's the region they're focusing on. So again, it's a watch this space as to, but it's all about, we found this effect, now we want to understand why this effect occurs, and again, how can it help drug design and everything else in the future.
I think I stopped listening to you after you said Korean chicken, John. No, I'm just kidding. I'm playing. No, that is, again, just really fascinating how they design these things and how they think about it and the complexity of it. It's not easy to try to figure out like how one person feels hungry, right? Like that's
and I definitely don't want a drug that stops me feeling hungry when I see a plate of Korean fried chicken. I mean,
I don't know. I'm torn. I wonder. Not only that, I wonder if it has some. It does seem like it might have. If it has that type of nausea to it, I hope there's no other, like, maybe other stresses that it could cause psychologically. But who knows? It's certainly.
In terms of the nausea thing, that the Roche, another company we haven't really talked about. They've got a GLP-1 GIP, so it's like two pathways they're trying to do, and that's what tirzepatide is. So it's slightly different from Wegovy, and Wegovy's pure GLP-1. I think tirzepatide's a GLP.
Yep.
But the Roche one, that's a major side effect for them. Is they've shown real promise in terms of weight loss, but in clinical they're struggling with the vomiting at higher doses. So, it's something I hadn't realized it was quite that much of an issue until I read these couple of articles. So anyway, shall we move on to how this impacts us?
Yeah, I mean, John, I certainly we have a number of ways to measure GLP-1 analogs, if you put it, but I don't know where you want to take us, because this comes.
Yeah, I mean, I can tell you what we can talk. Right? We can talk about our experience, and then obviously,
biology, right? Just start the biology of GLP-1, right? There's like a prolytic form, an active form, and a non-active form, right? There's like three different forms, or am I mistaken there?
I am honestly, you're talking.
Yeah, so like when the analogs are like, so just endogenously, I think the GLP, and you mentioned GIPs and stuff like that, but there are really, in kind of how maybe my simple mind, or at least from what I'm learning about it, and I'm by no stretch an expert in this, John. I know enough to be dangerous, but I do believe that, like anything in the body, a lot of hormones come in like a form, right? They're kind of inert, and then something happens and you knock off a little piece of it and it becomes active, and then when it's done doing its thing, it needs to degrade and go away. So there's always like three forms of stuff. So just to I think that becomes a key piece to we're going to dig into the GLP-1 analogs, but the first thing we got to take into account is the endogenous version of this. I think that's the hardest part. But we have a lot of platforms and we can dive in there. But I want to, that to me is still the. But you can talk about the power of hybrid mass spec here, but that's still to me. Correct me if I'm wrong, but the analog and the endogenous version are different. But I don't know. I feel like we've got a lot of experience measuring it by ligand binding assay. We've got experience measuring it by hybrid mass spec as well as LCMS. But you pick it up from here, John. But I can tell you, in the LBA world, man, it's a lot to ask out of a ligand binding assay. We're just looking for total GLP-1, right? It doesn't really matter. A lot of times we can't tell it unless someone's created a custom antibody. Then we can look at different versions of it, like endogenous. But otherwise, analog, or most ELISA kits for GLP-1 are going to pick up the analog and the endogenous version of it. But you go ahead, John.
I think, well, it's interesting while you're talking about this. So the news article that I was missing, because I had the wrong printout. Basically, so the headline is: Lasker Award goes to 3 scientists who pioneered research behind GLP-1 drugs. And so the three people are Joe Habner from Massachusetts General Hospital, Svetlana Moisov from the Rockefeller University, and both of them were responsible for figuring out some of the molecular mysteries of the natural version of GLP-1, which plays into what you were talking about. And then Lotte Bjerre-Knudsen from Novo Nordisk was honoured for figuring out how to turn the short-lived natural molecule into a longer-lasting drug, like we talked about earlier. Reading this, so Habner discovered what would be called GLP-1 by looking at glucagon genes from anglerfish. The reason being that anglerfish have an unusual organ that makes copious quantities of the blood sugar stimulating hormone, and so it meant that they could look at that before, because there were rules in place preventing looking directly at human genes. So he found that fish genes contained code not only for glucagon, but for a similar gene that looked like glucagon with 6 extra amino acids on one end. And that's what I think. So then the Moisov moved from New York to Boston to help disentangle how that molecular end cap on GLP-1 gets cleaved to release the active molecule. So there's your base and active versions, and it goes on further to say how they figured out its effect, etc. And like we touched on earlier, I keep forgetting her name. I'm really bad about this. Lotte Knudsen. She obviously led the team that figured out how to attach fatty acids to the drug and make them longer lasting. And the first one for Novo Nordisk was liraglutide, and obviously semaglutide came later. So yeah, it all plays into that. So from bioanalytical perspective, so these are, even with the fatty acid attached, these are molecular weights in the 3,000 to 4,000 range, maybe 3,000 to 5,000 range. So they're ideal for electrospray LCMS.
Yeah, the standard, right? If it's under 10, sorry, I want to make sure.
Yeah, absolutely. Yeah. So
by the way, John, I got to give one Ozempic. Right, the commercial. Sorry,
I've not seen the commercial.
Oh my God, it's everywhere. It's because you don't watch TV.
I just don't watch much TV.
Yeah, sorry, no. But because of the size, you can do intact. We call it standard LCMS is going to work.
Yep.
Right, let's dive into right.
So we can do standard LCMS, but and we have touched on this in the past, but it's always worth reminding people, because you've got basically two very different regions to that molecule. You've got the peptide, which we would consider more polar in nature, and what you've added by adding a fatty acid, you actually add this long lipid chain, which is very non-polar. So if we came up with an assay for a peptide, we take a particular approach, and we have what the chromatography is likely to, your chromatographic approach is likely to be, etc. You have clear expectations in terms of how it will behave in the chromatography and in the mass spec. But one of our enemies with LCMS is carryover, and having that lipid chain actually, well, first of all, it'll affect the chromatography, but the biggest issue for us is stickiness. It absorbs to surfaces and things like that. So carryover is a nightmare with these compounds. So we developed assays for liraglutide, semaglutide, all which worked. They were in the non-GLP group, but we did do a test where we have the hybrid team who can work, who mainly work with things above 10,000 molecular weight, but can look at these smaller molecules. If you can get a peptide that's representative, a smaller peptide that's representative of the bigger peptide, then actually you're losing that fatty acid chain, and you can develop an assay for these analytes where you're looking at using a surrogate peptide for the bigger entity, but you're away from that carryover issue because it becomes more predictable analytically because you don't have that fatty acid chain, which is is affecting how your analyte behaves in the system. So we have.
I think I followed you, John. I think that made sense to me.
Yeah, we haven't taken that any further than a proof of concept, but it's there, and it's certainly something we could jump back on. And because again, we don't know what other GLP-1s people are looking at in the future that may actually benefit from taking a hybrid rather than looking at it intact.
Because on the LBA side, there are I think MSD has a GLP-1 kit that we've got experience
Mhm.
with. There are other ELISA kits. I don't know if we've fully validated. I feel like we've done a number of studies. Like I said, it can measure total GLP-1. I don't. I don't. We should have maybe John. We could have looked at what that. Well, I don't know the sensitivity levels. I wonder if we could compare the
Well,
two.
that's what would be really
Yeah,
interesting.
that's
You-
shame on you and I. I don't know. I don't know what the levels are. I feel like they're nanogram levels in the kit that we have, but it could go into picogram. I feel like it's a pretty sensitive and robust kit.
Yeah,
I
and
would, I'd
that would be the interesting exercise if we had time and instruments to spare.
yeah.
Is to actually look at
And
how
John,
low can you go with LBA?
yeah.
What are the issues? How low can you go intact, and what are the issues? We know what those are because we've done a fair amount of work. And then how low can you go with a hybrid
Yeah,
approach and a high digestion approach?
yeah.
By using the surrogate peptide and see what issues are that way. We know we can do it by all three methods, but we just haven't done any real comparison of the three, which would be fascinating.
Yeah, you even now just gotta me thinking about that a little bit. Although I will say I can't emphasize enough that it's just total GLP-1 on the ELISA.
Sure. Yeah.
And a lot of times I feel like we've used the capture antibody from that for hybrid because we've got, this is a pretty hot space for us, John, right? And
Mhm.
that not only are there most of these, right, aren't just you have some methods that are straight up for the GLP-1, but a lot of them are like LNPs, right, where they're kind of packaged inside of something, or they're going to deliver the GLP-1 in some sort of um, gene therapeutic, right, John?
I think that's not where I've been. I think that might be more where you've been
Yeah,
involved.
and that's where we're those types of. So I guess you've done a good job of kind of just saying, hey, straight up LCMS. We've got methods coming out of our ears, right? All different species, right? Doesn't matter tissues, all things, right? You can do and you can look at I think of it as like the business end of the peptide with the kind of the stability end of the fatty acid chain. All of that can be done by
Yeah,
intact mass spec, right?
yeah, yep, yeah.
And it probably, and by the way, that if look, it's not we're not making recommendations, but if you can use that, use it. That's the most straightforward,
Mhm.
incredibly sensitive method to do it. There's nothing wrong with that.
Yeah,
What I'm about ready to dive into is more of those next generations of drugs where people are looking to kind of have, okay, what's my drug? I want to try to do it in such a fashion where it's a little more stable, and they got all sorts of intellectual property around some of this.
yes.
That's what it becomes into. They want to know, is it in an inactive form, an active form, or a degraded form? Because that can tell them the pharmacodynamic of it. See, it's just more
Sure.
than the PK.
Yeah.
Right,
Yeah.
it becomes we're scratching the surface at the bioanalysis, right?
Yeah.
We're not going to cover everything today, but if you just think of the PK piece, that is probably the easiest piece.
Absolutely, yeah, and so I would say if you're looking at the candidates coming through, the oral candidates, and then the more accepted GLP-1 analogues that are obviously the amylin derivatives, those are all easy by LCMS, and
Well, easy
mainly
is a
going
strong
into
word, right? Easy is you don't.
no, yeah,
We
easy,
have experience, and there's
exactly,
publications on it, but it's
yeah,
still, it's going to have to take some work, right?
sure, yeah, oh, totally,
Yeah.
totally, yeah.
And then flipping over, it's the endogenous versions that is a pharmacodynamic marker or a biomarker that we're getting asked a lot about. And that's where the power of hybrid might come in.
Sure,
Now you
absolutely.
can pull out GLP and then take a look at what the pieces are that you grabbed in that antibody, right? And that's some work there. There's feasibility studies. What antibody are we using? People have, again, some of this is proprietary as well. So, but it's really cool stuff, John. And then, the gene therapy piece. If someone's trying to knock in the gene, there's some molecular tests you can look at too. But we have experience with all of that. You start doing biodistribution. Can't emphasize enough some of that here, right? One of the articles touched on it. So you get to look at all sorts of factors there in terms of on target, off target. What are you trying to treat, right? Usually it's the liver, the pancreas, but you want to make sure you're not producing copious amounts of GLP in the testis, right? That can be bad. Those
Sure.
are the safety
Yeah.
markers. So just, I guess I'm off my soapbox for a second there, John. We kind of touched on the right technologies for the job. I hope we covered that a little bit there, right? Maybe not. I feel like we did, but then maybe you were going down the path of some of the challenges for the MS approach versus the hybrid approach, right?
I mean, I think we're confident that we can
like straightforward is the way to go, right?
produce an assay for any of the current candidates that are more conventional in design. We can easily support that, or readily support that. I think we have this final topic that you threw out there. This challenges for LBA, and tissue issues. So that's and when you're moving into the gene therapy piece, that's where I'm sitting back and becoming a spectator and listening.
Yeah, so just to make sure we round out our agenda here, right? We talked the mass spec is the way to go, and I don't know, John. There's the challenges there. You can maybe share. Yeah. I don't know exactly what they are, but I imagine we've ironed those out pretty seamlessly, given the copious amounts of GLP-1. It's
one of those things that the more you have experience with a class of therapeutics, the more you learn from it, and the more it sets you up for the future. So, in the initial days of working on the GLP-1s, there are challenges, like carryovers you can't underestimate. You carry over an absorption, that's a big deal, but you know how to deal with it. It's similar to what we see with the oligonucleotides. The more experience that we build with oligonucleotides, the more it becomes a routine entity to deal with, rather than like something that is a real challenge. They're still challenging, but you've built a bank of knowledge that if you see a certain thing, you have at least a few potential strategies as how you're going to tackle that. So we're well down the path with these kind of entities. So
and then when you think about hybrid, it really, the only time to me it gives value is if you are trying to discern a mutated version versus an endogenous, or something about, again, active, inactive, inert forms. And by the way, we're like in the throes of thinking about how to do that, John. Right? We haven't done that. That's what I'm saying. But I think it's fast approaching, and we're ready. Right? We got ideas. We got lots of ideas, and we got the hybrid mass spec team that can do it. And I'm sure, I feel like that's something maybe in another few months we'll revisit this. This is not going away, right? The industry experts say this is the number one field right now.
Oh, sure, absolutely, yeah.
So this is not something we're going to talk about this more, right? But I want but right now, if someone came to us, hybrid's it's not necessary unless, again, there's more pharmacodynamic is how I think of that, versus just straight up PK work on it.
Sure.
But then the LBA piece, that's where I mentioned there is, like, when people we're being asked to like look at a tissue in a preclinical setting and trying to discern if certain regions of that tissue have different expression levels, and that's it, the endogenous level of the tissue becomes challenging. That's the issue. So we can't discern using an LBA assay if we can tell the difference between the two. And so we're working with a proprietary reagent to do that. See, that's the key piece. We're in the throes of that, but and then the other thing is, when you cut a tissue, John, there's some stresses there.
Sure.
And so you can imagine trying to cut something, 5 sections of the same tissue, you can get some spurious results. And that is not, that challenge is not specific to GLP-1. That's a challenge to anybody who's trying to pull that much. Normally you just grind up the whole tissue, John.
Sure. Yep.
So to try and cut and say, is there a distal and proximal piece of the organ that that's asking a lot out of a ligand binding assay, and to be determined, right? And then again, getting back to, is it the endogenous or the therapeutic version of it? That's still
sure
TBD. But those are, that kind of rounds out our agenda, John. That's the challenge. And
that's where we're going with this.
LBA specific. That's what we're seeing today.
Sure.
It might get kicked over to our hybrid mess. I think the hybrid team's actually maybe taking a shot on goal here, and we're able to discern what we're supposed to do by LBA. But we're like in the middle of it. But it's just really great science around GLP-1, and it's all leading towards bigger, badder versions of it, right, John? Less side effects, less injections, maybe no injections, all that kind of stuff. But I don't know. Any parting thoughts, John, before we? No, I think you got a question for us too today.
It's one of those things that when when we suggested this as a topic, I was thinking, but we talked about this fairly recently. What more is a tad? But there is so much happening.
There has been, yes.
Yeah, there is so much happening, and like I say, they are interesting bioanalytical entities to deal with. So it is worth revisiting. It is a nice topic to talk about.
And then there's even, I think, some misinformation, John, right? There's misinformation out there about
sure.
GLP-1s.
Yeah.
And I'm not an online user, but periodically I've had a few people send me like, oh, is this true? And I'm like, it doesn't sound like it is. No. Some misnomer about what it could and couldn't do. But, I'm not going to perpetuate those things.
Yeah,
but I think there is a lot of stuff you got to take with a grain of salt about just how much weight you're going to lose. Mm-hmm. And certainly, we didn't talk. There are some side effects with nausea and stuff, but I think there's still some, just like any therapeutic, the long-term stuff is still a little bit, but I can tell you this, it probably outweighs it, whatever that side effect is, getting healthier and losing 20% of your body and getting your body to work better is probably, we got a couple, someone we know near and dear is on it, and they've lost 50 pounds. They're feeling better. They're moving better, golfing better. So,
it's interesting that, I think it was Blue Cross did the survey where they said, that a lot of users of, I think it was mainly semaglutide, gave up after around 2 years. So, but I think it was one of those. It was an interesting article to read, but there was still a lot of questions about the article, which is why I haven't touched on it. But it's something that hopefully we'll learn more about that in the future. Is that really a real trend or not? And again, it might be, does that come down to side effects, etc.? And if other forms don't have so much of the nausea side effect, for instance, maybe maybe they find that people stick with it more. I
think my brother stopped taking it because of that. But
it's interesting. I'll be honest, in terms of online stuff, my online presence is getting less by the week because there's so much nonsense floating out there that I think if I read something, you have to verify it from multiple different sources before it's real.
Even some of the bigger news outlets are susceptible to some of that. They'll print something and it's not right. And usually some really reliable ones too.
I'm very careful on stuff, and will. Like I say, I'll caveat as much as possible, particularly if I haven't found anything to corroborate something. I wouldn't go there. Anyway, that's enough on that
episode here, John. I guess before we dive into what our plans are and so forth, we want to go with that question we were asked, John.
Oh, sure, absolutely. Yeah, we do actually have feedback.
I don't know, Jeremy. What was asked, Jeremy? Go ahead.
So, Kayleen asks, is Dom sponsored by Uni? If not, he really should be. Every time I hear him mention them in these episodes, I get the impulse to get my Italian husband one.
Well, thank you. I am not, but it's funny you should mention that. I had my eye on upgrading because my Uni's a couple years old, and they've got some wonderful, much more just more ergonomical is how I'd like to put it. A little bit smaller footprint than the one I have. They got some features about it that mine doesn't have. So if anyone from Uni is listening, please do. I work for cheap, I'll beta test stuff for you. But no, I appreciate that type of question. And I can't go an episode without mentioning pizzeria, but I did make some. I fired it up recently, John, and my wife gave me an A+. I got the dough from Whole Foods this time. I think I got my new secret place to get dough from. Whole Foods has got some great.
Okay.
Yeah. And I made 2 batches of pasta sauce now. I've made like 40 quarts of pasta sauce. I've been a busy Italian, John. And my wife says it's some of the best I've ever made because I doubled the sugar in it. She likes it sweet.
Ooh.
Yeah, exactly. You went, ugh. Then you would have liked my more. I made a second batch that didn't have nearly as much. It's heavy basil and garlic. It's a little more.
Yeah, I'm not.
You like that one a little more, but John,
I don't need the sweet, but each to their own.
Yeah. It's just it's almost a pinch. It's borderline. It's not terrible. It's not like you probably haven't eaten Chef Boyardee in forever, but like
No, I've never. Don't give it.
but those are super. Any sort of like if you buy a Stouffer's lasagna or something like that, their sauce is super sweet.
Sure.
Kind of what she grew up on, so she likes it. Where I did not grow up on that.
Yeah, it's interesting. I think generally that's one thing as someone from the UK. A number of like those mass-produced American foods are far sweeter than we would ever expect. You know,
super sweet.
Yeah,
I think it's just like the glucose initiative that makes people eat, right? Like any Twinkies and cupcakes. But those are kind of going away a little bit, although Chef Boyardee is a lot healthier than mac and cheese. But we'll move on, John. Trust me, a box of mac and cheese is terrible for you relative to that. It's on the Eat This, Not That books from years ago. Boy,
sure. Yep.
So John, you can tell us about your weekend. You go first, or anything you want to.
So I this last weekend we just had my eldest son staying with us for a few days. It was his first trip here in 9 months, so it was good to catch up with him. And he spent a lot of time catching up with friends and stuff like that.
Nice.
Next weekend is going to be a quiet one because my wife will be flying. Oh, she's going to New York before flying to Denmark, so her travel is becoming much more interesting than mine. Although I'm sort of still, I would say I'm still coming down from the fact I spent 2 weeks in the UK, which was excellent.
Yeah, that's great.
At some point, I need to pull photos together and sort all that stuff out. But yeah, I had a brilliant time.
Well, I got
relatively quiet phase now.
Yeah, no, I got the one son. He went off to college, but he turned 19, so we got to see him this past weekend for his birthday. But John, you talked about it's weird, like seeing your son. It's different. It's really been. I don't know if I'm not really too emotional about these things, but it really seems like it's a different chapter in life. When we can't unpack the toothpaste, so to speak. He's not going to be the boy who used to live in the.
Sure. Absolutely,
and that's still trying to adjust to that. We're only 6 weeks in from that, or 5 weeks now, but we have parents alumni weekend, which it's not alumni, it's just parent weekend, and so I think there's an event on Friday night out in Lawrence at Kansas University with his fraternity, and so that'll keep us busy this weekend, and to get to spend some time with them again would be great. We were happy to take him out for lunch and sing him happy birthday. That was good. But
I mean,
on that, I don't have anything interesting going on, John. No cool trips, but other than working on making pasta sauce and gonna fire up my big green egg here and make some chicken thighs I got going on. That's this week's agenda. But no, it seems like all work and no plays making Dom a dull boy. It's been very busy working, and do have some cool trips coming up. We're going to AAPS. Well, I'm going to make a plug there. I'll be out, and if anybody's listening and you're going to the AAPS conference in Salt Lake City, that's the 21st, 2nd, and 3rd of October. I'll be out there. We even have a little event I think we're hosting, so come see us at our booth. There's my plug, John. Of course, we'll go to Spain. You and I.
Yep.
We'll be in
Barcelona. We'll be counting down to that. I need to book my ticket for that.
Yeah, we'll probably have another podcast or two before that, but
sure,
I am all locked in. I got my flight, I got my hotel, I got my Spanish. I'm practicing up. Yo practica mi español un poco para.
Well done, you.
But no, it'll be fun to be. I haven't been in Spain since 1989, John. Oh man, that hurt to say. That's funny. But no, any last parting thoughts here, John? Otherwise, we'll see you in the funny pages.
I think that's it. I think we wrapped up another one. Let's look forward to getting ready for number 82.